Welcome to another episode from the Veterinary Medicine Digest. Today, we are embarking on a comprehensive exploration of a complex and often under-recognized condition within the field of wildlife pathology, Neuroschistosomiasis in aquatic birds. To truly appreciate the intricacies of this disease, we must first establish a rigorous foundational understanding of the parasites involved, their evolutionary history, and the broader context of schistosomiasis as a significant global health challenge for both humans and animals. The family Schistosomatidae represents a unique and fascinating group within the class Trematoda and the order Strigidida. These parasites are obligate two-host organisms that demonstrate a high degree of specialization in their life cycles. They reside within the vascular systems of their definitive endothermic vertebrate hosts, which include both mammals and birds, while utilizing aquatic or amphibious gastropods as their essential intermediate hosts. Unlike the vast majority of digenine trematodes, schistosomatids are exclusively dioecious, meaning they possess separate male and female individuals, and they frequently exhibit marked sexual dimorphism. This evolutionary adaptation is believed to be a consequence of their life within the high-pressure environment of the vertebrate circulatory system. The muscular male is required to navigate against the flow of blood within the portal system, while the slender female is specialized for the deposition of eggs within the fine vessels of the intestinal wall. The historical depth of our relationship with these parasites is profound, as evidence of human schistosomiasis has been identified in Egyptian mummies dating back to 1250 years before the Common Era and in Chinese mummies from the Han Dynasty. In the modern era, schistosomiasis remains one of the most recalcitrant, neglected tropical diseases, affecting over 230 million people across 74 endemic countries. While human medicine has focused heavily on species such as Schistosoma mansoni, Schistosoma haematobium, and Schistosoma japonicum, the diversity of avian-infecting schistosomes is arguably even greater. Recent phylogenomic research utilizing targeted sequence capture of ultra-conserved elements has shed light on the complex diversification of this family. By analyzing hundreds of independent nuclear loci, researchers have identified that schistosomatids likely first appeared in marine birds and gastropods before colonizing freshwater environments and diversifying into the clades we recognize today. This evolutionary journey involved multiple host-switching events, including two separate captures of mammalian hosts. One of the most intriguing aspects of this diversification is the role of intermediate host switching, which appears to be a primary driver of speciation within the family. Schistosomatids exploit at least 16 different families of gastropods, a much wider range than other prominent trematode families like Fasciolidae. Before we dive into the specific neurological manifestations, it is important to understand the typical systemic pathology of avian schistosomiasis. In many avian species, these parasites cause significant vascular lesions. For instance, studies of Whooper Swans in Japan found that schistosomiasis was associated with a condition known as venous hypertrophy. This is characterized by a marked nodular proliferation of medial smooth muscle fibers, which often leads to the complete obliteration of the vascular lumen. These lesions are commonly found in the medium-sized veins of the mesentery and the various layers of the intestinal tract, including the duodenum, jejunum, ileum, and cecum. Furthermore, vascular damage can extend to the capsules and interstitia of the liver, spleen, kidney, and heart. In some cases, this condition has been described as obliterative endophlebitis, particularly in mute swans, where the vascular hypertrophy was attributed to intimal rather than medial proliferation. Similarly, research on Tundra Swans in North America identified infections of Allobilharzia visceralis, which also caused endophlebitis in the mesenteric veins. These systemic infections are often associated with emaciation and high mortality rates in wild bird populations. However, the focus of our deep dive today is the aberrant and severe manifestation of these infections within the central nervous system, a condition termed neuroschistosomiasis. In humans, neuroschistosomiasis is a well-documented, though underdiagnosed, complication where eggs or occasionally adult worms reach the brain or spinal cord. The pathogenesis involves an immunogenic interaction between the deposited eggs and the host's delayed hypersensitivity reaction. The myrichidium within the egg secretes proteolytic enzymes and glycoproteins that stimulate the formation of periovular granulomas. In human cases, Schistosoma japonicum, which produces smaller eggs, is more frequently associated with cerebral symptoms, while the larger eggs of Schistosoma mansoni are more often retained in the spinal cord. The eggs can reach the central nervous system through various routes, including embolization from the portal system via portopulmonary shunts, or the aberrant migration of adult worms through the valvulus vertebral venous plexus known as Batson's plexus. This context brings us to the pivotal retrospective study conducted by Sasaki and colleagues, which investigated neuroschistosomies in 12 aquatic birds from zoological and aviary collections between 1998 and 2021. This study is particularly significant because it represents one of the few efforts to correlate detailed histological findings with molecular species identification in avian neuroschistosomiasis. The birds included in this study were quite diverse in terms of species, including brown pelicans, harlequin ducks, nene geese, American flamingos, golden-eyed ducks, mute swans, ringneck ducks, red-breasted geese, Orinoco geese, and a great white pelican. The median age of the affected birds was 12 years, with all individuals being adults. This observation aligns with the understanding that older birds may have had repeated exposure to schistosomes, leading to the accumulation of chronic lesions. The clinical presentations of these birds varied widely, though most were found dead. Some exhibited clear neurological signs or general weakness before death, while others were being treated for unrelated conditions like fractures or renal failure. Interestingly, upon post-mortem examination, there were no remarkable gross findings within the central nervous system of any of the birds. This underscores the necessity of microscopic examination to identify the disease. However, systemic gross lesions were noted in some cases, such as nodular foci in the intestines or red linear structures on the duodenal mucosa, which pointed toward a broader schistosomiasis infection. The histological analysis revealed that all 12 birds had trematode eggs within the brain parenchyma. These eggs were often embryonated, containing a recognizable myrichidium, or they were in various stages of degeneration, including collapsed capsules or complete mineralization. The study identified 2 primary patterns of tissue reaction to these eggs. In 6 of the cases, the eggs were not associated with any inflammatory response or other tissue reaction. The researchers hypothesized that in these instances, the migration of the eggs to the brain may have occurred so shortly before death that the host's immune system did not have sufficient time to mount a response. In the remaining 6 cases, the presence of the eggs elicited significant inflammation. The most common pattern was granulomatous inflammation, which is highly consistent with the chronic phase of Neuroschistosomiasis seen in humans. In 2 cases, the inflammation was specifically lymphocytic in nature. A particularly important histological finding in 2 of the birds was the presence of trematode egg emboli within the blood vessels of the brain. In these instances, the mineralized or degenerate eggs were physically lodged inside the vessels without an associated inflammatory reaction, but the vascular walls showed a smudgy, eosinophilic appearance indicative of early damage. This finding provides strong evidence for the embolic route of egg dispersal to the central nervous system. Beyond the brain, 4 of the birds showed evidence of systemic schistosomiasis, with eggs found in the liver, kidneys, spleen, gallbladder, and various parts of the gastrointestinal tract, including the proventriculus and cloaca. In one remarkable case involving a harlequin duck, schistosomula or larval trematodes were actually identified within the mesenteric arteries. These larvae were characterized by their eosinophilic tegument and parenchymatous bodies, although they lacked reproductive structures at that stage of development. The defining achievement of the Sasaki study was the molecular identification of the parasites responsible for these lesions. Using polymerase chain reaction and sequencing of the internal transcribed spacer 2 region of the trematode deoxyribonucleic acid, the researchers were able to identify the parasite in several cases. Despite the challenges of working with formalin-fixed paraffin-embedded tissues, which can lead to deoxyribonucleic acid degradation, they successfully identified Dendrotobilharzia pulverulenta in 4 of the birds: the harlequin duck, the Nina goose, the red-breasted merganser, and the brown pelican. Dendrotobilharzia pulverulenta is a particularly interesting member of the Schistosomatidae family. While most avian schistosomes are found in the venous system, this specific disc is known for its tropism for the arterial system. It has been found in the lower aorta and femoral arteries of various diving ducks, including the genera Ithia, Bucephala, and Mergus. The researchers suggested that this arterial tropism might actually facilitate the travel of the eggs to the brain, as they would be following the natural direction of blood flow from the heart to the cranial structures. The molecular analysis further revealed interesting intraspecies variations within Dendrotobilharzia pulverulenta. Some sequences were identical to those previously recorded from common mergansers and snails in the United States and China, while others lacked a specific 6-base pair insertion or showed single nucleotide polymorphisms. These variations are consistent with what is expected across a geographically widespread species. The study's results suggest that Dendrotobilharzia pulverulenta may be far more common in avian Neuroschistosomiasis than previously realized, potentially having been underdiagnosed in the past due to a lack of molecular confirmation. When assessing the clinical significance of these findings, the study concluded that Neuroschistosomiasis was likely the cause of death, or at least a major contributing factor, in 5 of the 12 birds. In these individuals, the presence of significant granulomatous inflammation around the eggs in the brain was deemed a critical finding. In other cases, the neurological lesions were considered incidental compared to other severe conditions, such as systemic amyloidosis, rhabdomyosarcoma, or severe acute ovarian follicular rupture. However, even in incidental cases, the inflammation in the brain might have contributed to the general weakness or neurological signs reported by clinicians. The broader implications of this research are significant for veterinary pathology and wildlife management. The identification of Dendrotobilharzia pulverulenta in multiple species of aquatic birds, beyond the traditionally studied swans and geese, expands our understanding of the host range of this parasite. It also highlights the importance of incorporating molecular techniques into routine pathological investigations, as morphological identification alone is often unreliable due to substantial variation among conspecifics. Furthermore, the study emphasized that the duration of formalin fixation is a critical factor in the success of molecular identification, as prolonged fixation significantly reduces the sensitivity of polymerase chain reaction. This research also adds a layer of complexity to our understanding of how these parasites interact with their environments. As Ebbs and colleagues noted in their phylogenomic work, the ability of schistosomatids to switch between different snail and avian hosts is a testament to their evolutionary potential and adaptive flexibility. This flexibility is likely supported by high genetic diversity and large effective population sizes, particularly in parasites of migratory birds that encounter diverse habitats. Understanding these transmission dynamics is not only vital for the health of wild bird populations, but also for managing the zoonotic risk of sacarial dermatitis, or swimmer's itch, in humans who share these aquatic environments. In conclusion, the retrospective study by Sasaki and her team provides a vital window into the hidden world of avian Neuroschistosomiasis. By combining traditional histopathology with modern molecular tools, they have characterized a disease that is subtle in its gross presentation but devastating in its microscopic reality. We have seen how these parasites, with their ancient evolutionary roots and complex host relationships, can find their way from the arterial systems of diving birds into the delicate tissues of the brain, eliciting powerful immune responses or silently damaging vascular structures. This work serves as a reminder of the incredible diversity of the natural world and the constant, intricate struggle between host and parasite that shapes the health of our planet's ecosystems. Thank you for listening. The titles for the reference papers are in the video description. From VetMed Digest, where we keep minds inspired, hearts light, and tails wagging.